Hello, everyone. I'm Anas Al Ubaidi, and this is lecture 2, session 2 from chapter 19 of the Advanced Medical Science Liaison and Medical Monitor Certification course program by CCRPS. As the chapter is about the advanced monitoring strategies, and the topic for this session is about the advanced protocol development and risk assessment. Introduction. Advanced protocol development and risk assessment are the cornerstones of clinical trial success, serving as the foundation for scientifically valid, ethically conducted, and operationally feasible research. Meaning that in order for the trials to be conducted properly and appropriately, in order to reach the most accurate outcomes, then advanced protocol development and risk management assessment, both advanced protocol development and risk assessment are cornerstones, are essential, are the foundations in order to reach our goals or our goal by conducting any study, clinical trial study. The protocol acts as a detailed and binding blueprint outlining every aspect of a clinical study from objectives to methodologies, in other words, from A to Z. It ensures alignment among stakeholders by standardizing procedures, safeguarding participant safety, patient safety, and meeting the expectations of regulators like the FDA, Food and Drug Administration Agency, or EMA, the European Medicines Agency, or in Europe, FDA in USA and EMA in Europe or European Union in EU. A well-crafted protocol minimizes ambiguities and enhances the study's efficiency by preemptively addressing logistical and operational challenges. Risk assessment, on the other hand, serves as a continuous process that complements protocol development by identifying, evaluating, and mitigating potential obstacles that arise during trial execution. It anticipates issues affecting patient safety, data quality, and regulatory compliance, enabling sponsors and investigators to proactively implement corrective measures. Advanced risk assessment tools such as risk based monitoring, RBM, failure mode and effects analysis, FMEA, short for failure mode and effects analysis, failure mode and effects analysis, failure mode and effects analysis, and centralized real-time dashboards have revolutionized how risk is managed, ensuring trials operate with agility and precision. For advanced physician medical monitors, for MMs, proficiency in protocol development and risk assessment is critical. By mastering these skills, monitors ensure trials deliver reliable outcomes, comply with the global regulations, and prioritize an ethical participant experience. Now, the key points for the topic. We'll start with essential components of a clinical trial protocol. A clinical protocol serves as a framework that defines the trial's purpose, design, and operational steps by providing a roadmap from start to finish. Below are the critical components of an effective protocol. Core structural components. The components, despite of the order, we have general information outlines essential trial details such as the protocol title, version, dates, and contact information. For the principal investigator, PI, sponsor, and MM medical monitor. Example, evaluating efficacy of a drug as X in stage 4 lung cancer protocol version 3 June 2024. Background and justification explains the rationale for the study, including prior research findings and investigational product details. Example, citing preclinical and early phase data supporting dose escalation plans in oncology trials. Objectives and endpoints defines the trial's purpose through clear primary and secondary goals tied to measurable outcomes. Example, primary endpoint, reduction in HbA1c by 10% within 6 months in diabetes patients. Study design describes the overarching trial structure, including randomization, blinding, and intervention strategies. Example, a double-blind placebo-controlled design with a 2-to-1 randomization for a cholesterol-lowering agent. Participant selection includes inclusion and exclusion criteria, the eligibility criteria that will be listed in the protocol, recruitment strategies, and plans for withdrawal or follow-up. And all that mentioned will be mentioned in a study trial protocol. Example, excluding individuals with pre-existing kidney conditions for neurotoxic drug trials. Treatment schedules and procedures provides dosing regimens, treatment durations, and methods for drug administration. These contents also will be mentioned in the protocol. Example, administer compound A, 50 milligram QD for 6 weeks, followed by a 2-week washout period. Safety monitoring details methods for documenting AEs, adverse events, and serious adverse events, SAEs. Example, mandating weekly AE adverse event logs and reporting grade 3 toxicities within 24 hours. Statistical analysis plan outlines hypothesis tests, sample size justifications, power calculations, and data interpretation methods. Example, Kaplan-Meier plots for survival endpoints in cancer studies. Mastery techniques for protocol development. Develop modular protocol templates for efficiency and consistency across trials. Engage multidisciplinary teams, as for an example, statisticians, clinicians, and site staff to address factors like eligibility and feasibility. The key takeaway here, a clear, comprehensive protocol ensures alignment among stakeholders, mitigates risks, and minimizes ambiguities in trial execution. And here is a summarized diagram on the anatomy of a clinical trial protocol. As here we have the general information, background and justification, objectives and endpoints, study design, participant selection. Best practices for writing protocols. We have regulatory alignment, ensure compliance with the global standards like ICH E6R2, FDA guidelines or guidance in USA, and GDPR in European Union requires for data protection. Example, integrate GDPR compliant measures like anonymized data collection for EU-based trials to ensure participant privacy. Clarity and conciseness. Use precise language to communicate endpoints, timelines, and data collection modalities. Avoid overly complex language. Example is to replace, for instance, vague phrases like improved outcomes with 10% reduction in baseline tumor size over 6 months. Stakeholder consultation. Consult investigators, data managers, and other stakeholders to ensure the protocol is operationally feasible. Example, align stratification criteria, as for an example, by gender or treatment history with site investigators' practical capabilities. Iterative document development. Circulate draft synopses to address concerns prior to before finalizing the full protocol. Early collaboration reduces later amendments. Common pitfalls. Overloading settings with unnecessary details, especially for exploratory trials. Failing to consider operational constraints like limited investigator training or site resources. Key takeaway, balance through thoroughness with operational feasibility to maintain protocol clarity while avoiding overcomplication. And here we have a summarized diagram to show the enhancing protocol development. As here we have the regulatory alignment, stakeholder consultation, clarity and conciseness, and iterative document development. In order to end up, if we comply or follow all these components of the development, then we'll end up with effective protocol development. Other than that, we'll have ineffective protocol development otherwise. Advanced risk assessment methodologies. Risk assessment strengthens clinical trials by identifying, evaluating, and mitigating vulnerabilities across operations. Structured methodologies in risk assessment. We have SWOT analysis, SWO2, identifies project strengths, weaknesses, opportunities, and threats. As for example, strength here, experienced biometrics team, weakness, limited pediatric site experience. Failure modes and effects analysis. Failure modes and effects analysis, FMEA, failure modes and effects analysis, as predicts potential operational failures, assigning risk scores based on likelihood, severity, and detectability. As here, for example, highlighting the risk of treatment misallocation in multi-center trials and mitigating it through automated randomization algorithms. RBM risk-based monitoring prioritizes monitoring resources to high-risk sites while using centralized technology for low-risk ones. Example, using ECRF, electronic case report form data log, as a trigger for site-specific intervention. Visual risk tools use radar charts for graphic representation of risks. As for example, radar visualizations compare high probability versus low detectability scenarios among trial risks. Recent advancements in risk assessment automation, as dashboards track real-time protocol deviations or serious adverse event frequency. Example, oversight software flagged a site with delayed AE adverse event reporting, prompting immediate retraining. Central monitoring platforms, tools aggregate and evaluate all site-level risk triggers in real time. Key takeaway, a proactive approach using advanced technologies ensures trials remain efficient and compliant despite complex risks. And here is a summarized diagram unveiling risk assessment in clinical trials. As here we have risk assessment methodologies, we have the SWOT or SWOT analysis, failure modes and effects analysis, FMEA, risk-based monitoring or management, RBM, visual risk tools, recent advancements. Real world applications, we have the area technique example. As areas, we have protocol development, risk assessment, oversight, monitoring, adherence, and statistical safety reporting. The final takeaway here, master of protocol development and risk assessment provides advanced physician medical monitors, MMs, with the tools to lead effective and compliant clinical trials. By creating comprehensive protocols and implementing proactive risk monitoring strategies, advanced monitors can significantly improve trial outcomes while prioritizing participant safety, scientific rigor, and efficient operations. Leveraging both traditional best practices and emerging technologies will empower future clinical trial leaders to confidently address the complexities of modern risk landscapes. And here's a diagram on enhancing clinical trial outcomes, as here protocol development, we have risk assessment, proactive monitoring, trial outcome improvement, enhanced will end up with enhanced trial leadership. Summary tables, upcoming tables to summarize what we have covered through this lecture. This table on components of a clinical trial protocol, component description, example, importance, real-world impact. Components, we have general information, study objectives, study design, participant selection, statistical analysis, a plan. Another table on best practices for protocol writing, best practice description, example, key benefit, application, and protocol development. As best practices, we have regulatory alignment, clarity and precision, stakeholder collaboration, detail and study procedures, early draft synopsis. A table on risk assessment methodologies, approach, methodology, example, key strength, impact on trial management approaches. We have SWO2 SWOT analysis, FMEA, failure modes and effects analysis, risk-based monitoring, RBM, visual risk scoring, metrics-based monitoring. We have table on challenges in protocol development and risk assessment as the challenge, impact, proposed solution, example, outcome. Challenge: overloading with details, lack of operational feasibility, poor stakeholder consensus, detection of risks too late, missing key scenarios. Table on recent advancements in protocol development and risk assessment, advancement, description, example, benefit, real-world applications, centralized risk tools. We have automation and monitoring, adaptive protocol designs, wearables for remote data, metrics-based risk reviews. Application table, scenarios for each scenario, key principle applied, key actions, timeline, what to watch out for. As scenarios, we have drafting protocol for a cardiovascular study, evaluating risks in remote monitoring trials, addressing serious adverse event SAE risks in oncology trials, incorporating GDPR compliance in EU, European Union trials, designing inclusion criteria for pediatric trials, automated dashboard for serious adverse event SAE tracking, ensuring adherence to GCP, good clinical practice in global trials, rapid protocol adjustments during initial trials, identifying weaknesses at rural trial sites, and then evaluating efficacy endpoints in an immunotherapy study. Now, we'll use some MCQs, few multiple choice questions to revise what we have covered through this lecture. As question 1, what is the primary purpose of a clinical trial protocol? Is it A, to summarize team meeting agendas, B, to provide blueprints ensuring scientific validity, compliance, and participant safety, C, to replace standard operating procedures, SOPs, or D, to reduce trial costs through fewer participant criteria? So the protocol has nothing to do on team meeting agendas, has nothing to do with that. Has nothing to do with the SOP, as SOP is a completely separate documentation. And has nothing to do, has no role in reducing trial costs. So the answer would be B, to provide a blueprint ensuring scientific validity, compliance, and participant safety, and many more things for conducting the trial. As a protocol outlines the detailed design procedures and methodologies for clinical trials to ensure consistency, regulatory compliance, and the safety of all participants. Question 2. Which section of the protocol highlights the standards for monitoring adverse events AEs? Is it statistical analysis plan A, B, treatment plan, C, safety monitoring, or D, study objectives and endpoints? So it is C. The answer, as safety monitoring is including or linked to the adverse events AEs monitoring, so safety monitoring. As the safety monitoring section ensures proper documentation and management of adverse events AEs, safeguarding participant well-being using standardized criteria like CTCAE, for instance, as for an example. Question 3, what is a critical best practice when writing a clinical trial protocol? Is it using vague language for flexibility, B, obtaining input from multidisciplinary stakeholders, C, avoiding regulatory alignment to reduce complexity, or D, leaving procedural details open-ended? So it's not D, not to leave details open-ended. Everything has to be cleared, detailed and cleared. Regulatory alignment is needed, not to avoid, so not C. And not to use vague language, not A. So definitely it's B, as of obtaining input from multidisciplinary stakeholders from different background specialties in order to cover every aspect within developing a protocol or writing a protocol. As collaboration with investigators, statisticians, and regulatory experts ensures the protocol accurately addresses feasibility, compliance, and scientific rigor. Question 4, what does risk-based monitoring RBM prioritize in clinical trials? A, equal oversight across all sites. B, allocating resources based on risk probability and impact. C, eliminating monitoring for low priority trials. Or D, conducting 100% source data verification for every site. So it is B, as of allocating the resources, available resources, based on risk probability and impact. as RBM focuses on optimizing resources by targeting high-risk areas to improve efficiency and ensure data integrity while maintaining participant or patient safety. Question 5. Which methodology identifies potential clinical trial failures and prioritizes mitigation strategies? Is it SWOT, SWOT analysis, A or B, Kaplan-Meier analysis, or C, failure mode and effects analysis, FMEA, or D, visual risk scoring? So it is about the trial failures and to prioritize mitigation strategies. So it is C, as of failure mode and effects analysis, FMEA. C is the answer, as FMEA evaluates potential trial failures, assigning scores for the likelihood and impact of risks to ensure proactive risk mitigation plans are implemented. And here we come to end of this lecture. Thank you very much and goodbye.